Diabetes is the fifth leading cause of death. In 2018, around 405 million people worldwide were diagnosed with diabetes, and it's estimated that this number will elevate to around 510 million by 2030. Diabetes leads to a wide range of complications, including but not limited to blindness and cardiovascular disease. Life expectancy for people living with diabetes can be reduced by up to 20 years. Type 2 diabetes arises when pancreatic beta cells fail to secrete enough insulin to cope with increases in blood glucose. Various genetic and environmental factors increase the risk of type 2 diabetes. Obesity and a sedentary lifestyle are two of the major risk factors. Dietary interventions can therefore be used to help restore the responsiveness of beta cells. Carbohydrates, lipids, and proteins are the three types of macronutrients. They have all been shown to play important roles in regulating beta cell functions. Glucose is the primary driver of insulin release from beta cells. Different types of fatty acids are able to either promote insulin secretion or inhibit secretion indirectly via reducing beta cell health and survival. Some branched-chain amino acids can stimulate insulin secretion, but they may also hamper glucose metabolism. However, the molecular mechanisms by which these macronutrients modulate insulin secretion in human beta cells are still being investigated. How do we decide which one or combination of these three macronutrients should be used in a dietary treatment if our goal is to carefully control insulin secretion? If we could figure out the molecular pathways by which carbohydrates, lipids, or proteins stimulate beta cell function, then based on the genetic profile of a patient with type 2 diabetes, we could personalize a dietary treatment that could optimally reverse that patient's conditions. In the Johnson lab, islet samples from 80 donors were exposed to glucose, fatty acids, or amino acids. Unpublished results have shown that insulin secretory responses to glucose, fatty acids, and amino acids vary widely between individuals. It is possible that these variations are due to the genetic and environmental exposure differences between the individuals that integrate at the level of the beta cell proteome. Based on these findings, my current objective is to study the proteomes of the human pancreatic beta cells from these 80 samples using mass spectrometry. From the mass spectrometry data, I will first look at the abundance of various proteins involved in the insulin secretion pathways. Then, I will try to correlate the different protein levels to the existing nutrient-induced insulin secretory data. If we could identify the proteins that may predispose the beta cells to release different insulin levels to different macronutrients, namely carbohydrates, lipids, and proteins, the levels of these proteins in a particular individual could then help us personalize an optimal diabetes dietary treatment for that person. Thank you.